Thank you so much for taking the time to join the Arthritis National Research Foundation's fourth in a 5-part webinar research spotlight series. I'm Emily Boyd-Storman, the CEO of ANRF, and along with our board of directors, staff, and those participating in the panel, I'd like to welcome you to the spotlight on ankylosing spondylitis. We're so grateful for your participation and support and your interest and your time. Moderating the discussion today is the chair of the ANRF Scientific Advisory Board, Dr. Craig Walsh. In addition to chairing the SAB, Dr. Walsh is also a professor of molecular biology and biochemistry at UC Irvine and co-director of the Institute for Immunology and director of the Multiple Sclerosis Research Center. Before we start the webinar, just 2 important pieces of information to note. 1, the webinar is being recorded and we'll have it available on our website at a later date. 2, if you have questions, please type them in the Q&A and we'll try and get to all of them. If we aren't able to get to yours, please email us at info@curearthritis.org after the webinar and we'll get back to you. Now let's move on to the presentations featuring the impressive work of our ANRF scholars and guests. So without further ado, I'd like to turn it over to Dr. Walsh.
All right, well, thank you, Emily, and I just want to welcome everyone for showing up today to hear about ankylosing spondylitis and some of the really interesting work that's happening. In particular, we're going to be talking about ankylosing spondylitis, which in some related diseases. Ankylosing spondylitis is a type of disease that we call an autoimmune disease. It has certain autoimmune components associated with it. And when we talk about autoimmunity, basically what we're talking about is where our immune system kind of goes out of control. So normally our immune system is there to protect us from, pathogens such as SARS-CoV-2 that's involved in causing COVID-19. And normally that works fine. It also protects us from cancers and other things. But every once in a while, there are certain errors and mistakes that happen in our immune system. And in particular, certain classes of immune cells called T cells that can cause real problems. And one example of that is ankylosing spondylitis. So today we'll be hearing different presentations having to do with this kind of autoinflammatory and autoimmune disease. And I'm not going to I don't want to steal any thunder from our speakers today, so I'm not going to get into too many more details about it. I will just say my own background. I study autoimmune disease, primarily in the context of the central nervous system. So we work on multiple sclerosis in my laboratory. More recently, we're also looking at the role of T cells in Alzheimer's disease. Okay, so without further ado, I want to introduce our first speaker, which is Ruth Napier. And Ruth is an ANRF fellow, so she is currently funded by the ANRF, and we're certainly delighted to have her presenting today. So she's coming to us from Oregon Health and Science University, as well as the VA Portland Health Care System. And her work is really kind of focused on innate immune signaling. And so I'm sure you'll get into this, Ruth, but when we talk about innate immune signaling versus adaptive immune signaling, humans, just like all vertebrates, we have both. We have an adaptive immune system that allows us to be able to respond to certain very specific molecules that exist in not just in pathogens, but also tumors, and those can cause us problems when we have these autoimmune diseases. But the second class of immunity is present in a whole host of different organisms, all the way from very, very simple microbes and parasites and invertebrates. And that is called innate immunity. Innate immunity responds to what are called pathogen-associated molecular patterns. These are molecules that are basically present within microbes that allow them to do what they need to do to survive. So without further ado, I want to introduce Ruth, and she's going to present a talk entitled From Human to Mouse and Back, Understanding Disease and Ankylosing Spondylitis. Ruth?
Thank you so much for that introduction. Let me pull up these slides. So, okay, can you guys hear me?
Yes.
Great. Thank you again for this introduction, and thank you so much for inviting me to talk about my research. And thank you, ANRF, for making the studies that we're about to hear about possible. With that said, today I'm going to focus my talk on ankylosing spondylitis, as Dr. Walsh was talking about. This is a type of inflammatory arthritis that is fairly prevalent with 1.4% of the global population. It primarily affects the spine, and I have pictured here a healthy spine next to a spine that was taken from an individual who had severe ankylosis, which means fusion of the spine or the vertebrae. And you can see to the right, this is an X-ray of the spine of an individual with ankylosing spondylitis. So this disease, as Dr. Walsh was saying, is considered an autoimmune disease. And its onset is fairly early, around the age of 20, and seems to predominate in men. So what causes ankylosing spondylitis? The short answer is we don't know. But what we do know is based on a lot of genetic work that's been done over the years. So I'm sure most of you are familiar with the protein HLA-B27. So 90% of patients with ankylosing spondylitis are positive for this protein. However, the general population, there's about 3% of us that actually have the HLA-B27 protein, but only 1 to 5% of those people actually ever develop ankylosing spondylitis. So these data suggest to us scientists that, trying to go to my next, sorry, that there's something more to it. And so further studies were done in the genetic world, and we found that there was a whole host of genetic loci that were actually correlated with more innate immune responses, including this gene CARD9, which I'll be talking about today. And so here I've pictured the CARD9 mutation, but in addition to other mutations that might influence the development of ankylosing spondylitis, we think there's a huge environmental component. So today I'm going to focus my talk on CARD9. So CARD9 is actually a very interesting protein because it's actually a microbial sensor. And so when you as a human are exposed to environmental fungi, so molds, yeasts, both pathogenic and just environmental, the CARD9 pathway is activated. So I've zoomed in here on a human cell here in purple. And so when you normally, a healthy person or encounters environmental fungi, this binds to a fungal trigger receptor, which activates the protein CARD9 inside your cells. CARD9 then activates a whole host of genes that are expressed from your DNA, some of which some of you have probably heard of, which are pro-inflammatory cytokines. So these are inflammatory molecules that activate other parts of your immune system. And we know that when the CARD9 pathway is activated after a fungal trigger, that this causes the development of pathogenic T cells, which Dr. Walsh was alluding to as one of the primary causes of ankylosing spondylitis. And that naturally this pathway was put in your body as thought to protect you against fungal infections. So the question my research has is, if you have a mutation in CARD9, why does that make you more susceptible to developing ankylosing spondylitis? So to ask that question, I began in a mouse model of ankylosing spondylitis. So the mouse model that I have is genetically predisposed to developing ankylosing spondylitis, but prior to developing it must be exposed to a fungal trigger. And so here I've pictured yeast. So if you expose this mouse to yeast, it develops an arthritis and a spondylitis, so an inflammation of the spine. So to ask the role of CARD9 in this mouse, I developed this mouse, but I knocked out or deleted the gene CARD9. So you can see in our normal genetically susceptible mice that after being exposed to fungus, that they develop a chronic and sustained arthritis. And here at 8 weeks past fungal trigger, you can see an ankle with clearly developed peripheral arthritis. Whereas when I knocked the gene CARD9 out of these genetically susceptible mice, they were completely protected against development of the disease. I'm further able to look at disease pathogenesis using a more molecular technique. This is called near-infrared imaging. And so what you're looking at here is the ankles and knees of these genetically susceptible mice. And so these mice have the gene CARD9 and these mice don't. And you can see here, the more fluorescence is the more inflammation that you have. And so mice that are lacking the gene CARD9 are again fully protected against any signs of inflammation on a molecular level. and also a clinical level. And here I had spinal cords that were taken from these mice. And so the mice that lacked the gene CARD9 didn't develop any spondylitis. So going back to our mouse model, so we have our genetically susceptible mouse. When they're exposed to yeast, they develop a CARD9 mediated arthritis and spondylitis. We know this because when we knock out CARD9, they're not able to develop the disease. And through further experimentation, we were able to find that CARD9 was actually functioning in a type of white blood cell in your peripheral blood called a neutrophil. In fact, when we were able to knock out or delete neutrophils in this mouse, that we were able to mimic that CARD9 deficient mouse. And so both CARD9 deficiency and neutrophil depletion both abrogate any signs of disease, suggesting that neutrophils are playing a really important function in the disease ankylosing spondylitis. So my next question was, how are neutrophils playing a role in disease in humans? So moving from the mouse model back into the humans, I activated a host of amazing collaborators, one of which you'll hear next, Dr. Liron Caplan. We're able to draw blood from patients with ankylosing spondylitis and then isolate neutrophils from their blood and do experimentation. And so one piece of data I wanted to show you that was done in collaboration with Dr. Caplan and Dr. Sen was we were able to look at almost 1,500 spondyloarthritis patients, which included ankylosing spondylitis and psoriatic arthritis patients, and how many neutrophils they had and how and if it correlated with disease severity based on BAS-Phi index. So we found that the higher the absolute number of neutrophils were in these patients' blood, that the higher disease score that these people had, suggesting that neutrophils, the more neutrophils contributed to worse disease. We're also able to look at patients who are treatment-free because we wanted to make sure that the treatment wasn't the thing that was increasing neutrophils, and this correlation still stood true. So what comes next in our research? I first wanted to go back over the power of this translational research approach. We started with a question that was relevant to human health, asking about why CARD9 mutations might provoke ankylosing spondylitis. We're able to then develop a mouse model to test and ask this question. And then hypotheses that were generated in the mouse model were then tested on human samples. And so now that we have these this evidence that neutrophils are important in humans, we're going to go back to our mice to ask more mechanistic questions. And then based on those assays, we're going to go back to the human samples and ask if that's what's powering those neutrophils in AS patients. And that would lead us to new therapeutic targets and whatnot, well, we would hope. So overall, just in summary, I showed you that CARD9 expression in mice and genetically susceptible mice actually drives disease. And so when you knock out CARD9, you block disease, and that this is functioning in mice through a neutrophil. And then when we went into humans, we were able to find evidence that neutrophils are positively correlating with disease in this model. So our next question is, well, how is CARD9 working in humans, and exactly how are the neutrophils changed? And with that, I'll thank my lab and all of the AS patients who've donated blood to our study and donors for Nrf for making this possible, and our other funding agents. So thank you.
All right. We're going to ask some questions for Dr. Napier. And first off, I want to start off and just ask, you guys are finding that neutrophils are involved. Oftentimes, those are associated with a special kind of T cell called a Th17 cell. And I'm wondering if you guys have looked to see whether there might be an involvement of Th17 cells and whether there might be an idea about eliciting antigens that could be involved in that.
That's a great question. And actually, that is aim 1 and aim 2 of my ANRF grant. And we are very excited about that. And you're exactly right. CARD9 is known to induce the Th17 response. And so it makes a lot of sense. And we actually have looked through genetic data, and individuals who have the CARD9 mutation, it's actually a gain of function, suggesting that CARD9 might be overactive, which would mean that it's theoretically pumping up those IL-17 producing cells. So yeah, right now we're looking in humans and mice to see if neutrophils directly or indirectly trigger that response.
All right. let's see, any questions from the audience?
No questions at this time, but if you do throughout the presentation, please feel free to chat them or enter them into the Q&A, and we can get to them at the end of the presentations as well.
All right. Well, thank you, Ruth. And I just want to say real quickly that Ruth is a fellow of the ANRF, and just kind of talk a little bit about the what the ANRF does very quickly before we move on to the next presenter. So the ANRF is primarily engaged in funding careers. So people that are either senior postdocs or starting out in their own laboratory as a faculty member somewhere. And I myself was an ANRF recipient. It was actually the very first grant that I got when I first started my lab here at UC Irvine. So it's had a big effect on me personally. And so I've been obviously really interested in trying to help the ANRF in that mission, because it's a very crucial stage during a scientific career. All right, so with that, let's move on to our next speaker. Aurora, Colorado.
Had a substantial amount of technical difficulty, so hopefully we're past that. Actually, let me see. I'm trying to share the screen. It's up online, but let's see.
Yeah, green button at the bottom of your screen. There you go. Perfect.
Got it. And let's see, I may have to switch the. How's that look?
Perfect.
Perfect.
Great. I apologize. My computer crashed literally at 17 minutes ago. So I just managed to pull this up. So
no worries. It's looking good. We've all been there during this crazy time. But thank you again for joining us.
Yes, and thank you to Ellie and Dr. Walsh and the board of NRF for asking me to present. I'll be presenting on clinical studies of axial spondyloarthritis. And again, my name is Ron Caplan. I'm the director of the University of Colorado program to advance spondyloarthritis treatment. These are my disclosures, none of which are relevant to the current presentation. And so what I'm going to cover in today's presentation is the general concept of axial spondyloarthritis, as well as the development and classification of spondyloarthritis. And then I'll show some problems and issues in spondyloarthritis and how we've used a resource known as the Program to Understand the Long-Term Outcome in Spondyloarthritis Registry, or PULSAR. And I'll just quickly skim over some study results from that registry. So as Ruth expertly conveyed, spondyloarthritis is a group of diseases that's characterized principally by involvement of the axial spine. And by the axial spine, we mean the main spine, as well as the sacroiliac joints in the pelvis. It can also present with peripheral arthritis, inflammation of the tendon and bone junction, known as enthesitis, as well as dactylitis, which is known as sausage digits, swelling of the entire digit, and skin and eye inflammation. And here we have a gentleman that has the characteristic posture of spondyloarthritis with a straightened back, as well as inflammation at the junction of his Achilles tendon, where it inserts on the bone of the calcaneus. So development of classification of spondyloarthritis involves three major phases or stages. The first, what we refer to as a preclinical or at-risk stage. Next, a earlier or milder stage known as non-radiographic spondyloarthritis, which is not a particularly great term, but that's the term that's been used. And then a later or more severe term known as ankylosing spondylitis, or sometimes referred to as radiographic spondyloarthritis. And we think in the preclinical phase that there are genetic susceptibility loci, the most famous of which are the HLA-B27, combined with environmental exposures that render a person at risk for the subsequent stages. Those first begin with inflammation, and then there's a reparative process that involves the generation of fatty tissue, which in certain instances can result in abnormal bone formation. I mentioned HLA-B27 as the genetic susceptibility loci, or the genetic marker that is most prominently featured in this disease. It's present in about 8% of the population, and about 10% of folks with that HLA-B27 will progress to actually developing the disease, and that occurs in the young adulthood. So I've arbitrarily picked at 25 years, that 10% progression. About 1% of the population ends up having axial spondyloarthritis, and those numbers should make sense based on what I just mentioned. About 50% of the folks with the axial spondyloarthritis, which is the broader category that includes both the earlier and milder disease, as well as the later and more severe disease, about 50% of those will develop the more severe disease at about 10 years, and they'll progress to what we know as ankylosing spondylitis. And depending on what phase a person's at, that has implications on both diagnosis and treatment, with things such as HLA-B27 testing done through the blood, physical exam maneuvers, and then imaging, various imaging modalities. MRI used in the early disease during a fatty repair to pick up the fatty repair process that's ongoing, and then CAT scans and X-rays for later disease. But there are problems along each of these stages, problems of diagnostic accuracy and delays in diagnosis. There are problems of how to assess inflammation and how to do that accurately. There are problems assessing the severity of disease when it comes to things such as CAT scans and X-rays. And then there are problems determining the best treatment. So one way that we address these sorts of problems is by bringing together resources. And the resource that we've developed over the last few years is PULSAR, the Program to Understand the Long-Term Outcomes in Spondyloarthritis. This is a registry, meaning that patients are collected from around the country, and they contribute to the same sort of data collection at every site. And we currently have about over 1,300 patients that are contributing from the following sites. And these registries can be used to address the clinical questions that were raised in the slides before. What is the best radiographic technique for imaging the sacroiliac joints? That's one possible question. And so we looked at the question of whether the addition of oblique radiographs help in the assessment. And what this basically means is that you could either take one X-ray, known as the Ferguson view of the pelvis, or a 3-view of the pelvis, which includes these oblique angles. So it's 3 X-rays versus 1. Now, the potential problem with this is that it could be more radiation in getting these additional views. However, it also might allow for more consistent information for the provider. And these were the results. I don't mean to belabor the details here, but in essence, you can see that the gray bars are higher, meaning that a higher percentage of folks met the modified New York criteria, which are the criteria that we use to say that a person has ankylosing spondylitis. And you can see that the gray bars are always taller, representing 3 views versus 1 view, which is the gold bars. You can see that the correlation across the different severities that we see on the X-rays from 0 to 4 also change. And so we took from this study that even though the dogma, the current thinking is that 1 radiograph is fine, and most providers ascribe to that view, we actually believe that you gain additional information by doing a 3-view, and so we advocate for that approach. Another question might be how clinical radiographic reports compare with radiologists' reading of the X-rays. And so we developed what we call a consensus reading, 3 different rheumatologists that together scored the radiographs. And we compared that to the scoring that's done in a clinical report. And we saw better correlation with a genetic marker, HLA-B27, and a higher percentage of folks meeting the classification criterion when it was when we depended on the rheumatologist's reading. And so this raises the question of what we can do in improving the quality of our clinical reports that are being read by radiographs. It also suggests that that rheumatologists should really be taking it upon themselves to read the radiographs themselves, and that's the approach that we advocate. Another question that we asked is how different TNFs, which are agents used, TNF inhibitors, which are agents used to treat spondyloarthritis, how they might behave in ankylosing spondylitis. And so we looked at how long patients maintained that therapy, and you could see that among these different therapies, the different versions of the TNF inhibitors, that different percentages of patients over time remained on them. And so, the etanercept and infliximab had the highest retention rates, and the lowest was seen with golimumab. And so it points that maybe there might be some preference, at least for groups of patients, between these agents if the goal is to keep patients on a drug longest rather than having them flip through drugs. And then lastly, as it was alluded to by our collaborator, Ruth, we have studies in which we use these registries to collect biologic samples and then collaborate with basic scientists. And one of the studies that we've done here, which Ruth presented some of the data for, looks at the percentages of different types of cells and how those correlate with traditional markers of inflammation. And so, these didn't perform fantastic, but there is a correlation between these different percentages of cells as represented by something called the neutrophil lymphocyte ratio, as well as the platelet lymphocyte ratio, with a traditional marker of inflammation known as the CRP. And so we're developing ways of how we can take existing labs that are being drawn already to monitor medication safety and repurpose those to monitor disease activity in patients and therefore have a better handle on the disease. So I hope in going through this brief presentation that I've conveyed the following points: that spondyloarthritis is a group of diseases characterized by arthritis of the spine and the sacroiliac joints, that there are many issues with understanding who develops these diseases, how to make a best diagnosis, how to monitor patients, and how to treat them, and that we can leverage things such as the Pulsar registry to allow us to answer these questions in a collaborative scientific endeavor. Thank you very much. I will plug, and I'll leave the slide up here. I'll leave this last plug for patients that or family members of patients with spondyloarthritis. This is the contact information for our study that's being done at the University of Colorado, which is very similar to Pulsar, except that it's, instead of being based at the VA, it's at the university. And if you'd like, if you happen to be in the Colorado area and you'd like to volunteer for the study, again, as someone with the disease, a family member of someone with the disease, or someone who just wants to contribute, then these are the contact information. And I appreciate your attention.
All right, great, Dr. Caplan. I just have a quick, very basic question. Is there anything known about what makes somebody predisposed to developing axial spondyloarthritis as opposed to, or ankylosing spondylitis as opposed to, say, rheumatoid arthritis? I mean, a lot of the mechanisms look sort of shared between the two diseases, and yet it seems like it's much more localized in the case of the latter.
Yeah, there's certainly a lot more work that's being done in rheumatoid arthritis. I think that the field in spondyloarthritis is running about 10 years behind where it is in rheumatoid arthritis. And so the question that you ask is extremely pertinent, and it's why we have these programs such as PEERS, where we're looking at folks that don't have it, but maybe are family members of folks with spondyloarthritis and are therefore at higher risk. And our goal in doing that, collecting samples from patients like that, is to get them before they have disease and see what changed. That being said, areas that are that might be contributing include different types of pathogens, germs in the gut that might be contributing, and so there are certainly environmental exposures that might be different. And then genetically, of course, HLA-B27 is a risk factor for developing spondyloarthritis that's not present in other forms of arthritis. So we think there's definitely a different genetic background, and there are almost certainly different environmental exposures.
Now, with respect to the HLA-B27, I think that you mentioned that there's a certain proportion of folks that show up that have that obviously they don't develop the disease. I'm wondering if that has to do with the fact that there's just a huge amount of polymorphism and lots of other genes. If you go to different parts of the world and different groups of folks, whether there might be a refinement in understanding about the genetic loci, particularly with respect to MHC polymorphism?
Sure. Yeah, there have been, while HLA-B27 is clearly the predominant genotype, there have been other genes that have been linked, including ERAP1, a number of other genes, although they seem to contribute a lot less. And I think our understanding is rather, and our approach, quite frankly, is fairly simplistic at this stage. And so I think there's a lot of complexity in gene environmental interactions that we don't yet appreciate, and it's going to take huge efforts really around the world. And so we're certainly participating in those to try to answer those questions. Even the concept of spondyloarthritis and frankly, all autoimmune conditions is a very simplified notion, right? You know, spondyloarthritis, not every patient will have peripheral arthritis or inflammation in the eye. And so each patient is really quite unique. And while they share some pathophysiology, some genetics and things like that, that there's an incredible complexity that's being reduced down when we use terms like spondyloarthritis. And that can only be addressed if we have really large population studies that are very well powered to address these questions of individual patient variability.
All right, thank you. Let's see, do we have any other questions from the panel or from the audience?
I had kind of like a question comment, but about
Dr. Napier.
Oh, sorry, about the HLA-B27 comment. I mean, individuals who are part of a family who have AS patients or spondy patients in them, even if they're HLA-B27, the heritability is somewhat low, but if they have a first generation relative, it gets higher and higher and higher. And so I think like emphasizing that this is truly like a polygenetic disease, and like the HLA-B27 is probably the baseline susceptibility. And that if you were to actually profile families and look for like, what's the secondary, like it may be ERAP, it may be CARD9, but actually I mean, some of the stronger data, if you go back to the genetic studies, is things like CARD9, that like more people with CARD9 develop the disease than just all over people who have HLA-B27. So I think there's still a lot to learn. That's all I wanted to say.
Sure, thanks, Dr. Napier. And I would imagine also, as was alluded to, that there are environmental factors as well as is often the case for these kinds of complex diseases. Any other questions for Dr. Caplan before we move on? Emily, do we have any questions coming from the attendees?
No questions in the chat, but we'll keep monitoring, and if any come through, we'll check in at the end of the next presentation.
Great. All right. Well, thank you very much, Dr. Caplan. Appreciate it. We're going to move on to the next speaker. And this is Michael Paley, who's an MD, Ph.D. physician scientist and instructor of medicine in the Division of Rheumatology at Washington University in St. Louis School of Medicine. So Dr. Paley is going to present a talk entitled Understanding Ankylosing Spondylitis Through the Eye.
Thanks very much, Craig. Dr. Caplan, if you could stop sharing your screen, that would let me share my presentation. Thanks. So thanks very much for that introduction, and pull that up. Okay, thank you very much for attending and listening to the work and allowing me to be included in this incredible group. So I run the ocular rheumatology clinic at Washington University, and one of the a common patient that we see or is referred to us is, for example, a 30 or 40-year-old woman. She goes to see her ophthalmologist because she develops a red and painful eye, and her ophthalmologist diagnoses her with anterior uveitis, or inflammation in the front part of her eye, runs some lab tests, and one of which comes back positive for B27, right? So the ophthalmologist says, oh, you have a positive B27, so you need to go see a rheumatologist. So when they come and see me, she's has some back pain and stiffness, and we get x-rays that show some damage and inflammation in her sacroiliac joint in her pelvis. And so diagnosing her with ankylosing spondylitis is pretty straightforward. What becomes more challenging for us is when she asks, well, my HLA-B27 test was positive, so are my kids going to get this, which has been alluded to just 2 minutes ago. And we don't have the tools clinically to help stratify whether this is a high-risk person or a low-risk, family member because of the combination of genetic and environmental factors. But this is what motivates a lot of the work. And so we think we take an approach that's a little bit unique in terms of understanding ankylosing spondylitis by trying to understand what's going on in the eye. And that's because both anterior uveitis and ankylosing spondylitis, or the axial spondyloarthritis, are strongly linked to HLA-B27. So not only is there a genetic linkage, they often occur in the same individual. So here on the graph on the right, you can see over time, this is time since, as you move from left to right, time since your diagnosis of ankylosing spondylitis. And as you can see, as more in years past, on the y-axis, this is like the frequency of people that have had at least one flare of anterior uveitis. So as time passes since your diagnosis, you're more and more likely to develop a flare of anterior uveitis, so that over the course of your lifetime, about half of all individuals have at least one flare of anterior uveitis. So because both of these types of inflammation in the eye and in the spine are genetically linked to HLA-B27, and they often co-occur in the same individual, we think that understanding how anterior uveitis works will help us understand how ankylosing spondylitis occurs. So we also think there's a couple of advantages to studying the eye compared to some of the other tissues based on the anatomy. So ideally, when you're studying a disease and trying to find what are the bad actors in the tissue, you would go and you would sample that tissue directly to see who's there and what they're doing. So this is challenging when you're trying to sample the sacroiliac joint because of anatomical reasons, and this sampling is not commonly done clinically, and it's even more rarely done, if at all, for research purposes. And that's somewhat in contrast to the eye. So here's a cartoon of the eye on the right. So the front of the eye, this is where light comes in and it lands on the black part, the retina, which then sends signals to the brain so you can see. So when you get inflammation in your eye or anterior uveitis, the tissues that get inflamed are your ciliary body and your iris. And when this happens, you have a lot of immune cells that come in from the blood and enter these tissues and participate in the inflammatory process. And when they exit these tissues, they actually can enter into this space called the anterior chamber. And the cells start floating around like little snowflakes in a snow globe. And so by studying these cells, we think we can infer what's going on in the tissue itself, since they just emerged from this inflamed tissue. So you can actually sample the cells with a very small syringe, both clinically for diagnostic purposes as well as for research purposes. And you can get out about 2 to 4 drops of fluid, which contains several thousand cells. So previously we weren't able to do very much with that limited material, but with the onset of new technologies like single-cell RNA sequencing, we're able to generate a fair number or a large amount of data from just a very small amount of tissue. And so you might be thinking, well, what types of cells are we looking for by this analysis? So to do that, I'm going to just take a step back and describe a little bit of what HLA-B27 does in the whole family of HLA molecules. And that is to show off what's going on the inside of the cell to the rest of the immune system. So lots of cells. So cells make lots and lots of different types of proteins, one of which is cartooned here as this chicken. And at some point in the lifespan of the protein, it ends up in this machinery called the proteasome, which chops up the protein into little pieces called peptides. And these peptides get loaded onto these HLA molecules on the surface of the cell to be shown off to the immune system, where for example, the sheriffs in town, the CD8 T cells, patrol through the body, looking at all these different peptides on the HLA molecules to make sure all the cells are healthy, right? So different molecules, different HLAs will do the head of the chicken or the bone or the thigh or the wing. And the whole point of this process is so that when cells get infected with a virus, these viral proteins go through the same process and get chopped up by the proteasome into little peptides and then loaded onto the surface of the cell, so that CD8 T cells can recognize that this cell is infected with a virus and can eliminate the cell in order to contain the infection. So that's what happens in sort of the appropriate healthy response. But what's thought is that HLA-B27 is one of many different HLA B alleles that you can get from either parent. And it's thought that this specific B27 may present a peptide that's somewhat unique and activate a certain set of CD8 T cells that may be contributing to the disease process in terms of either developing the disease or promoting inflammation. So what those CD8 T cells might be, however, is not really clear. And there's a ton of different flavors of CD8 T cells, so lots of different sheriffs in town. And they all come with their own sort of markers or, colors or proteins that are on the surface that, annotate or connote different functions. But what we've identified preliminarily, at least, is a flavor of CD8 T cell that expresses this molecule KLRB1 that we think is relevant for uveitis in the context of HLA-B27. So if you sample cells in the eye versus the blood of patients with uveitis that don't have HLA-B27, you don't see a very high frequency of this type of CD8 T cell that expresses KLRB1. Whereas if you look at individuals that have uveitis that do have HLA-B27, there's some frequency in the blood, but you can see there's a really high concentration of cells in the eye, such that in this early analysis, about half of all CD8 T cells in the eye are this flavor of CD8 T cell, which is highly suspicious for us and suggests potentially contributing to the inflammation that goes on in the eye and potentially in the spine as well. So our plan with the support of the Arthritis National Research Foundation is to sample a slightly larger set of individuals, both in the eye and the blood, to identify the cells that are in most abundance in the eye in these individuals, and then identify these candidate cells and then look in the blood in patients that are, in individuals that are healthy or individuals that have uveitis or ankylosing spondylitis to see which of these cells might be in higher frequency or concentration in the blood, or if they're more activated and more likely to contribute to inflammation because they're irritable. And the idea would be that defining these bad actors based on sampling the inflamed tissues can lead to an earlier diagnosis and hopefully better treatment outcomes. So with that, there's a lot of people that support this work, particularly in rheumatology, my mentor, Wayne Yokoyama, as well as my ophthalmology colleagues, especially Lynn Hassman, and obviously the support from the Arthritis National Research Foundation. But last but not least is the very generous patients that help contribute to this work. So with that, I'd love to take any questions.
All right, thank you, Dr. Paley. And just to point out that Dr. Paley is also an Nrf fellow, and certainly great to hear from him. I just have a couple of quick questions. So first of all, the KLRB1, that's part of the NK receptor family. What is known about what that can bind to and how that might influence CD8 T cell function? Yeah, so it's a homolog or ortholog of NK1.1 in mice. So it's part of the, like you said, the NK receptor family. It has mixed data in terms of whether it's an activating or an inhibitory receptor, whether it so if it's engaged on the surface of the cell for a T cell, sometimes that makes the T cell more activated and release more pro-inflammatory cytokines. Sometimes it makes it, in other contexts have been reported that it leads to less pro-inflammatory cytokines like TNF alpha. And so it's probably, in the context is, it's context dependent, like that makes a difference. There's several ligands that have been suggested or identified, one of which is LLT1, but I think the one that would be relevant in the eye is not really established or clear yet, or in the spine. The other question that sort of kind of comes back to your really nice little diagram, little cartoon of antigen presentation through B27. And so, you have this sort of chicken, and you've got some parts where, you might have, say, the drumstick, and other parts where it might be the breast or whatever. I mean, so I guess my question kind of comes back to this figure. When you're thinking about uveitis versus ankylosing spondylitis, do you think it's the same part of the chicken? In other words, the same peptide antigen that's driving the response in both sites, or do you think they're different? And if they're different, is there something about HLA-B27 that leads to, specifically expression of those antigenic peptides that leads to this sort of susceptibility to both of these autoimmune diseases?
Yeah, that's a gets to the heart of the matter. That's a great question. So I think, when HLA-B27 was first identified being associated with these diseases in the 70s, I think most people were thinking that, this presents a peptide in the eye or in the spine that's very specific to those tissues, and that's kind of why you get inflammation there. I think, in the 40 years plus since then, it's been very difficult to identify that peptide. And so I think the jury is still out on whether it's the same peptide. It also could be as, important that the environmental factors or the bacteria or the fungi that are in the gut may produce, they also have proteins that can lead to peptide synthesis that could stimulate CD8 T cells in the gut, and that would cross mimic, could activate them there, and they may be recruited to the spine or the eye for other reasons. So I think that's actually one of the proposals as part of what I want to do with the support from the ANRF is actually try to identify the peptides that can be recognized, because we can actually, with single-cell RNA sequencing, we can identify the T cell receptors that recognize different peptides in the eye. And hopefully we would make some progress towards that. Absolutely.
And then maybe compare those with the TCRs that are present in the spine, for example, right? Just to see if there's shared TCR sequences.
Right. If you have that data, yeah, absolutely.
Cool. Okay. Let's see. Any questions for Dr. Paley?
No questions on my end. Any questions from the other presenters? I want to make sure that you feel free to ask questions as well.
Yeah, absolutely. All right. Okay, well, why don't we thank Dr. Paley, really interesting work. And let's move on to our next presentation. And this is from Dr. Tejpal Gill, who's an assistant professor, the director of basic research for rheumatology in the Division of Arthritis and Rheumatic Diseases at Oregon Health and Sciences University. And the title of the talk is Gut Microbes and Spondyloarthritis. All right.
Thank you so much for the kind introduction, Dr. Walsh.
Thank you for joining us.
Okay, can you see my bigger slide or the presenter view?
We see both, so. There you go. That looks
Okay.
good.
Okay, thank you so much for that. Good morning, everyone. I'd like to start this presentation by thanking the Arthritis National Research Foundation for this opportunity to share our work. Today, I'll be discussing the highlights of our research on the role of gut microbes on spondyloarthritis. Since the presenters have already discussed about this, I'm not going to go into too much detail, but suffices to say that spondyloarthritis is basically an umbrella term that encompasses multiple different disorders, such as its prototypic ankylosing spondylitis, acute anterior uveitis, and the gut inflammation associated with these diseases. And they have a common genetic component, the HLA-B27. And these different disorders actually have common clinical symptoms, which could be articular or extra-articular. And bone-related symptoms can be inflammation in the axial skeleton, dactylitis, or abnormal bone formation. And on the extra-articular manifestations, you can get inflammation of the eye, the gut, and the skin. Okay, so why do we suspect gut microbiota, and why did we start looking about gut microbiota or gut microbial changes or dysbiosis in this disease? For the last decade or so, we've been studies in spondyloarthritis, and as you can see, there's an overlap between gut spondyloarthritis and inflammatory bowel disease. And gut inflammation has been very common in various forms of spondyloarthropathies, with AS patients having subclinical gut lesions, and some of them will go on and develop inflammatory bowel disease. On the other hand, 50 to 80% of IBD patients will have arthritis. Another factor that kind of evidence that came into light was the experimental models. They're highly dependent upon the gut microbiota for disease development. If you rear these animals in germ-free conditions, you would see, despite the presence of HLA-B27 transgene, you would not see any gut inflammation or arthritis in these animals. So the question that I asked almost 9 years ago when I started this microbial research in spondyloarthropathies is, does HLA-B27 drive alterations in the gut microbiota? And here I'm showing you the data from gene expression of two different backgrounds of rats, the Lewis backgrounds and Fisher backgrounds. These animals, upon overexpression of the HLA-B27 transgene, they have an increase in the inflammatory response. And that inflammatory response, which is marked by interferon gamma, type 1 and type 2 interferons, TNF alpha, IL-17 and 23, overlaps between these two backgrounds. And it's interesting that all of these inflammatory markers come up because blocking them has been basis of many biological therapies for spondyloarthritis. And this is the immune response. When I started looking into the gut microbiome, however, there's a different picture. Here I'm showing you the microbes which are dysbiotic or are differentially abundant in HLA-B27 transgenic rats as compared to the wild type rats. You can see that both these backgrounds have totally different microbes with only two microbes overlapping. And so how is that the immune response is common, but the microbes are different? So we performed a correlation analysis, and what we found was that different microbes in these backgrounds actually correlate to the common immune response. Furthermore, we went on and looked into the metabolic pathways that these microbes contribute to. And in both cases, in both backgrounds, Lewis and Fisher rats, we see common metabolic pathways being perturbed in association to inflammation. These could be the downregulation of really good or anti-inflammatory pathways such as the butanoid or propanoid. These are short-chain fatty acids, which are known to be anti-inflammatory in nature. Or there could be an increase in the inflammatory pathways, such as biosynthesis of LPS or, bacterial chemotaxis. And just proof of a principle in another separate experiment with these rats, when we gave, in this graph, with the gut inflammation plotted on the y-axis and wild type and B27 rats here, you can see when we gave these rats propanoid or butanoid in their drinking water, their levels of inflammation came down. So they have an anti-inflammatory role. And whereas the rats that were not given any of these inflammatory molecules had a higher level of gut inflammation. So furthermore, we wanted to see in all of these microbes, which are the ones which are driving the disease, because the microbial populations can change because of the inflammation and not because of the genetics. So to kind of separate the microbes which may be driving the disease, we took to research that came out a couple of years ago by Noah Palm and group, where they saw that in the gut microbiome, some of the microbes are coated with an immunoglobulin called the immunoglobulin A, and not all microbes are coated equally. So when they took only microbes that were coated with immunoglobulin A and transferred them into a mouse, they saw colitis, but the microbes that were not coated with this immunoglobulin A, upon transfer, they did not cause any disease. So we wanted to see if there's difference in the relative abundance of these immunoglobulin coating in patient populations. So switching gears, I'm showing you patient data. This is fecal samples from patients with axial spondyloarthritis. And here on this plot, the dark blue shows the microbes which are IgA coated in axial spondylitis patients, and the light blue shows the microbes which are uncoated or depleted in IgA. And as you can appreciate, there are many more microbes which are IgA positive in axial spondyloarthritis patients as compared to the healthy individuals shown here in black. And when I look up and search these individual microbes, I found that many of them have been associated with inflammation in either spondyloarthritis or inflammatory bowel disease. So indeed, the IgA coating of bacteria is increased in patients with axial spondyloarthritis. When we took this step forward and start looking into the metabolic contributions, okay, these microbes are either present in higher abundance or they're more coated. What are they doing in terms of their function? So when we look into the metabolic pathways, the IgA-coated metabolic pathways are, again, associated with perturbation of either anti-inflammatory metabolism, so the metabolism is decreased, or there's an increase in pathways that have been associated with inflammation. And interestingly, no such pathways have been found for the IgA negative fraction. So it kind of tells you that the IgA positive fraction of these microbes is driving the disease in patients as well. Now, this interaction between host and the microbe is so interesting, but the tissue where it happens, that's the mucosal tissue, is really, really hard to get. So what we are trying to do instead is we're trying to generate organoids, which essentially mean that they are microscopic gut, which has multiple types of cells, which mimic like a gut environment in a petri dish. So we're trying to generate, we have already generated, in fact, organoids and monolayers from HLA-B27 negative controls, and then patients with ankylosing spondylitis, as well as HLA-B27 positive healthy individuals. And what we are trying to do is we're trying to look into the barrier disruption by looking into markers for tight junction proteins. We're also aiming to understand host microbe interactions by giving them specific microbes and kind of, managing that interaction and looking closely in one-by-one terms. And I'm also looking into the sensitivity to microbial components such as LPS. And before I wind up, I just wanted to give you a little bit flavor of what the future implications of this microbiome research is. Although you might be familiar with fecal microbial transfer, which, as the name suggests, is you take the fecal microbiome from a healthy individual and give it to a patient, and that good microbial community can help in ameliorate the disease. However, there are many unknowns with fecal microbial transfer, since all of the microbes in the gut have not been characterized, and then there are viruses and fungi. So to kind of solve the problem and not put anything unknown in the patient, because we don't know how it's going to react, the synthetic gut microbiome is an alternative approach that's coming up. And what it means is culture the known microbes, which are known to have good effect on the gut, and give it to the patient in a known quantity and percentage. So there is no unknown here in terms of the microbes that we're giving. Another thing that came up just a couple of years ago in a paper, it's called Bespoke Bacteria. And what it means, it's the culture, it's a tailor-made bacteria, which you genetically engineer to produce antimicrobial peptides, which are essentially peptides against pathogens. And that way you can give it to the patients and kill the bad bugs. Or you move away from the microbes and give them microbial metabolites such as butyrate. So in the end, I would like to acknowledge all my present and past mentors, colleagues, and members of the Rosenbaum Gill Lab, as well as my funding agencies, the SAA and the MRF from OHSU. Thank you so much, and I'm ready to take any questions.
Thank you, Dr. Gill. Need to be a little bit careful because we want to be cognizant of the time, but I just have a quick question about the influence of treatment. So if some of these patients are on, say, anti-inflammatories, does that influence the microbiome in sort of a chicken and the egg kind of question?
Yes, thank you for that question. Actually, there has been a paper that I think recently came out from Matt Brown's group. And what they gave is they gave patients the TNF inhibitor, and then they started looking into their microbial community. And what they found was the patients who are on biologics, their microbial community after, in a longitudinal study, after they've been put on biologicals, their microbial communities look much more similar to the healthy individuals. And so it does not answer the chicken and the egg situation, but it just says that inflammation and microbial community, they are linked together, maybe too tightly spatially, temporally to be dissected, at least at this point. But definitely biologics treatment has shown, at least in few studies, to kind of bring back or restore homeostasis in the microbial community.
Great. Well, thank you very much. And I'm going to turn it back over to Emily.
Thank you so much, Dr. Walsh, and thank you so very much to all of our presenters today. It was a wonderful presentation, and great to hear from our scholars and our guests on all of the wonderful work being done in this area. Please join us in 2 weeks for our next webinar, spotlighting general autoimmunity. That will be held on November 17th at 11 o'clock Pacific Standard Time. And thank you again for your time, and we'll talk to you soon.
Thanks, everybody.
Thank you, everyone.